Stanford University.
Okay, so we will pick up on the one topic that was not covered from 2 days ago because you guys needed to go play around with these cellular automata first. So I will work with the assumption that everybody here has now spent 48 hours playing with those, but presumably because of the sleep deprivation you have forgotten much of it by now, so we will cover some of it. Okay, back to that issue of fractals and butterfly effects, and that whole business that by the time you look at chaotic systems that are determinist but aperiodic, all of that, when they seem to be lines crossing, getting back into the same spot, look closely enough and they're not going to actually be touching. And the centerpiece of why that matters was that whole business of, you know, these both appear to be the same, and take them out a decimal place and they're actually different, and a gazillion decimal places out. And the entire sort of rationale for thinking in that way is the notion that a very small difference here can make a difference one step to the left. And a million decimal places out there, a small difference will make a difference one before that. In a scale-free way, a fractal, this here, a difference here a million decimal places out, is just as likely to have consequences for 1 over as this one for 1 over, fractal, scale-free, all of that. But the critical thing that is encompassed in this is the notion that tiny little differences can have consequences that magnify and amplify into a butterfly effect. So cellular automata are a great way of seeing this principle along with a number of others that are relevant to all of this. Okay, so we start off with the very first one, and this is the one that you no doubt first discovered as a pattern, which made you deeply happy. And if you follow the rules, it was starting, there was this, which way is this facing? Starting at the bottom. Okay, so starting at the bottom, and what you see is these very simple rules, and out of it emerges a whole complex pattern. And we'll be seeing shortly the features of this that perfectly match what the requirements are for emergent complexity. But we'll see as the elements are lots of constituents, lots of building blocks, the building blocks being very simple. They're binary, either they are filled or not filled, extremely simple rules as to how the next generation gets formed. And in terms of the extremely simple rules, none of the rules have anything to do with other than the next generation. It is all local rules built around what the neighborhood is like for each one of these. So you put it together and out come these very structured patterns like these. And this is great. This is very exciting, except this isn't what you usually get. In most of these cellular automata systems, where you start off with an initial condition and a simple set of local neighbor rules for how you get reproduction into the next generations, in most cases, the patterns stop after a while. In the vast majority, they stop, they hit a wall, they go extinct. Aha! Two terms that I've already stuck in here that are biological metaphors start to seem less metaphorical after a while. First off, the notion that going from here to here to here to here represents each next generation, and the notion that, as we were just now, that the vast majority of these cellular automata systems go extinct. They fail after a while. So it's a very small subset. What you then also see is, in some ways, the critical point in this whole business is the relatively small number of starting states that succeed produce a remarkably small number of mature states that all look very similar to each other. In other words, you can start with a whole bunch of different conditions, and you will wind up with a bunch of smaller number of stereotypical patterns. Half of the cellular automata that wind up taking off look something like this with this pattern. What are we seeing? Convergence. Convergence, the notion that you can start with different forms and they will converge over time. What's this? Well, you just proved you look at the mature form and you can't know the starting state. The other thing is, starting at the beginning, just looking at this line, there is no way you can tell what it's going to look like 20 generations from now. You've got to march through it. In other words, the starting state gives you no predictive power about the mature state. This is a nonlinear system. The cellular automata encapsulates this business that most of these go extinct. Only a relatively few number of mature forms exist. It shows convergence. Very different starting states can converge into the same sort of patterns, and minor differences in the starting state can extend into very different consequences. It shows, in other words, butterfly effects. Okay, so appreciating this a bit. So what we did was then go to example number 2, where we changed the starting state just a little bit here. We shifted around some of the boxes. And what you see is something that looks roughly the same, but it's not exactly the same, but it's the same general feel to it. So that's great. But then we started an exercise of starting off with the initial boxes. This goes that way. The initial boxes, evenly spaced with one space between them, and apply the rules from there, and this is what you get. Totally boring, static, inorganic, inanimate. This is what it does for the rest of time. What this exercise then did, going to number 4, is what if we now space 2 boxes between each one of these? And here we have an extinction. This is one of those where it hits a wall and all the next lines are empty. Okay, how about 3 boxes in between the starting states? Okay, another form of extinction. Okay, how about 4 boxes between the starting states, and suddenly something very dynamic takes off. Applying the same rules, and all you've done is change the spacing between the starting states. And look at, for one thing, how close this was to going extinct up there on top, how asymmetrical the pattern is that comes out, and this particular one will stay asymmetrical forever. The ways in which that generated something very unexpected. There is no way you could sit there a priori and say, 1 box in between generates something that looks inanimate. 2 boxes, not going to work. 3 boxes, yeah, somewhere around 4 boxes in between. That's when dynamic systems suddenly take off. There is no way to have known that before without marching through this and actually seeing. Starting state tells you nothing about the mature state. Then we space it even further, and what we get is something similar again. This one is symmetrical. It is somewhat different from the previous one, but it's the same sorts of patterns that come up over and over. So what we've seen here is, by starting state, minor differences, big divergence between going extinct versus being a viable pattern. Minor differences in starting state, big divergence between symmetrical and asymmetrical patterns, tiny differences, butterfly effects. Okay, next, looking at the consequences here of introducing some asymmetry from the very beginning. The one on the left, up on top, has 4 boxes and 4 boxes. It has 8 boxes. The one on top has 8 boxes on the left, and the one on top on the right, just adding in 1 extra box on the side, so it's 4 and 5, adding a little asymmetry, and what you see is a very different pattern. And one of the things you tend to see in these pseudo-animate living pattern systems is starting states of asymmetry produce more dynamic systems, more dynamic patterns than even symmetrical ones. That's one of the only rules that comes out of there. So we're seeing now minor little differences producing major different consequences, divergencies, butterfly effects. Now showing this in a different way. And what we've got here are 4 different starting state conditions. The one on the far left is in fact the one from the previous one, the 4 and 4. 4 different starting state conditions where they're not enormously related to each other, the first one against the other 3, but the other 3 have minor differences. And the whole thing is 2 of these are identical after the first 20 generations or so, this one and this one. The 2 of them are identical, and for the rest of the universe, they will produce the same identical pattern, and looking at the mature state, you show up on the scene somewhere halfway down, and you could never ever know what the starting state was. Did it start like this, or did it start like this? A convergency here. And in this case, it's another one of those rules. Knowing the starting state doesn't allow you to predict the mature form. Knowing the mature form, you don't know which particular starting state brought it about, and the only way to figure it out is to stepwise go through the whole process, because you can't just iterate by a blueprint. There's no blueprint. Finally, the last one was giving you, instead of different starting boxes, in each case with the same reproduction rule, the last one was the same starting pattern of boxes with different, slightly different reproductive rules. And what you see here are totally different outcomes, depending on which variant. We have the beloved one on the top left, and you see here, by slightly changing the nearest neighbor rules, if and only if there's one neighbor with this property, if and only if there's 2 neighbors, if, and working through that way, and you see remarkably divergent outcomes. For one thing, you see that the majority of them produce something very boring, either boring extinct or boring repetitive in a very undynamic way. Only a small subset produce lively, animated, living systems. So we're seeing a whole bunch of biological metaphors here over and over and over, which is these starting states. You don't know the mature, you don't know the starting state. The generations, very simple rules for generations going one to the next. What we see also is the vast majority go extinct and produce either go extinct or some repetitive, very boring, crystallized type structure. A small subset, a tiny subset, produce instead dynamic patterns, and knowing what the starting state is not going to give you any predictability whatsoever of is this going to produce a dynamic pattern or not, nor does it allow you to look at a bunch of the starting states and say those 2 are going to produce the same mature pattern. And these are all properties of the evolution of different living systems. So you begin to see, okay, cellular automata, do you see some of these principles? The simplest level out there in the natural world is looking at all sorts of shells, seashells and tortoise shells by the seashore and whatever's, and they all have patterning on them that is derived from that first cellular automata rule, producing patterns that look a whole lot like these. And go online and look for them because I didn't get around to it in time, but producing all sorts of patterns in nature. Very common ones. What does that tell you? Very simple rules for generating the same complex patterns and different starting states, cellular automaton properties here. Another thing in a living biological system that begins to suggest this. Okay, so I do this research in East Africa, and every now and then over the years I've gone to this mountain called Mount Kenya, which is on the equator. It's about 17,000 feet. It's got glaciers up on top, so this is an equatorial glacial mountain. And you go up to about the 15,000 foot zone, and there's like it's moorland, almost everything is dead up there from the cold, and there's basically only like 4 or 5 different types of plants up there. Oh, already only a very small number that survive in that environment, and each one of them is very bizarre and distinctive looking. There's one of them that looks like a little like rosebud thing, except it's about 5 feet across. And then there's another one that has sort of a sprouty thing like this, and then a big central cactus-looking thing that isn't really cactus. So there's a few of these really distinctive, bizarre-looking plants. And in some way or other, that's what it takes to survive up there. So I have this friend who does research up in the Andes, and he does botany stuff up there, and he goes into this one range there that is on the equator and high enough that there's glaciers up there. A glacial equatorial mountain on the other side of the globe. So one day I'm sitting around and looking at some of his pictures there, and suddenly I look and say, that's the exact same plant. That's the big rosebud plant that's in Mount Kenya, and oh my God, that's the tall, sprouty one, and say, it's the exact same plant. How can that plant be over there? And we go rummage around in his botany, taxonomy stuff, and they're completely unrelated plants. They are taxonomically of no connection whatsoever, but what they've done is converged onto the same shape. And in some mysterious way, if you're going to be a plant growing on the equator at about 15,000 feet, there's only about 4 or 5 different ways of appearing. There's massive convergence, and there's only 4 or 5 ways that you could survive an environment like that. You get organisms in very dry environments, and there's like only 4 or 5 ways that you can go about being an organism that's super efficient at retaining water. And those are the only ones you see amongst desert animals, and completely unrelated ones have converged onto some of the same solutions. There's only a very finite number of ways to do legs and locomotion. 2 is good for weirdo things that fly have 6, creepy things have 8. You don't find 7. You don't find 3. You find some of the solutions here are immensely different starting states and have converged. What we see is in these living systems over and over stuff that look like cellular automata, where slight differences magnify enormously, butterfly effects, where you are modeling living systems in a very real way. Most of them go extinct, divergence, convergence, and where each one of these you get the smaller number, reflecting the fact that there's only a limited number of ways of doing rainforest, temperate zone rainforest in the Pacific Northwest. There's only a limited number of ways of doing tundra in Miami Beach. There's only a limited number of ways. In all of these, this convergence, and always reflecting that these are cellular automata. Okay, so hopefully you are now feeling desperately regretful that you didn't spend the last few days doing this, because these are so heartwarming. If you want to read a book that nobody in their right mind should read, it's a book by this guy named Steve Wolfram, who's one of the gods of computers and math, and was one of the sort of people who first developed cellular automata, and by all reports probably one of the largest egos on the planet. And he published a book, self-published it a few years ago, which he can because he is grotesquely wealthy from some of his computer programs, and just showing what a low-key sort of humble guy he is. He called the book A New Kind of Science, just showing that he wasn't just going from some little piddly new way of viewing the world, but here was his new type of science. And the book is about 1,200 pages, and I suspect not even his mother has read the thing. It is so impenetrable, and it sold a gazillion copies, and almost all of them are sitting in people's garages now, wading down drain pipes, because no one can actually read this thing. But an awful lot of what the book is about are patterns in nature coded for by very simple local rules. And the simple fact of that, you've got a lot of very smart people doing the cellular automata stuff, and they can't come up with rules where you could look at something beforehand a priori and know this one's going to survive, this one's going to go extinct, those two are going to turn the same, these two that differ by a slight smidgen are going to turn out to be enormously different. There's no rules for it. And the book has all sorts of cool pictures of the cellular automata looking things out in nature, and go buy it for somebody's birthday and see if they're not grateful for the rest of their lives. But his whole argument there is these show ways in which you can code for a lot of the complexity in the natural world with small numbers of simple rules. This whole business of emergence. This sets us up now for beginning to look at some of the ways in which we hit a wall the other day, ways in which the reductive model of understanding the universe stops working after a while. One version being the problem of not having enough numbers of things, not having enough neurons to do grandmother neurons beyond Jennifer Aniston, that whole business that you simply don't have enough neurons to do that beyond just the rare ones now and then. And what do you get instead? What has the solution turned out to be? This field that people focus on now called neural networks. And the point of neural networks is that information, again, is not coded in a single molecule, single synapse, single neuron, one neuron. This neuron knows one thing and one thing only, which is when there's a dot there. Instead, information is coded in networks, in patterns of neuronal activation. And just to give an example, and this is one that's in the Zebra book, and anyone who was in BioCore, I do this one, and I do it because I, at one point, learned the name of 3 Impressionist painters, except they're not coming to mind right now. Okay, so you've got 2 layers. Here's what a neural network would look like, a 2-layer one. These neurons on the bottom are boring, simple Hubel and Wiesel type neurons from the other day, where each neuron knows one thing and one thing only. This one knows how to recognize Gauguin paintings, this one recognizes Van Gogh, and this one Monet. Okay, each one of them is, obviously there's no Hubel and Wiesel neuron on Earth that's like that, but just for our purposes, they now project up to this next layer. Note, this neuron projects to 1, 2, and 3. This to 2, 3, and 4. This to 3, 4, and 5. So what does this neuron know about? This one knows how to recognize Gauguin. It's only getting information from this neuron. It's another one of those Hubel and Wiesel type, I know one fact and one fact only. This one here is another one of those. What does this neuron know about in the middle? That's the neuron that knows how to recognize Impressionist paintings. That's the one that says, I can't tell you who the artist is, but it's one of those Impressionists. It's not one of those Dutch masters. It's an Impressionist painting. And this one does it because it is getting information that is not available to these guys. It is getting information at the intersection of all these specific examples. These ones, number 2 and 4, those are ones that recognize Impressionist paintings also, but they're not as accurate at it as number 3 because they've got less examples to work off of. This is how a network would work, and what that suddenly begins to explain is something about the human brain versus a computer. Computers are amazing at doing sequential analytical stuff, like you get calculator things inside Cheerio boxes that can do more things than the human brain can do computationally, but what we can do is parallel processing. What we can do is patterns, resemblances, similarities, metaphorical similarities, physical similarities, and that's why you need networks like these. You don't need neurons that know one fact and one fact only. You need neurons where each one of them is at the intersection of a whole bunch of other inputs. Okay, example. So now suppose you've got a network, there's one neuron which fires, and there's a whole bunch of neurons sort of sending projections into it, and this is a neuron for remembering the name of that guy. What was the name of that guy? That guy, he was that Impressionist painter. So suddenly your Impressionist painter network is activating and firing at this neuron, so it's sitting there. So this is, now you've got your whole Impressionist network that's activating. What was the name of that guy? He was an Impressionist painter. He painted women dancers a lot of the time. So people who painted dancers, but it wasn't Degas. Okay, so it's not Degas circuit going in there. And what was that guy's name? God, I had that 7th grade art teacher who loved this guy's work. If I could remember her name, I would remember his name. Oh, remember the time I was at the museum and there was that really cute person who seemed to like it, and I had to pretend I liked this guy also, and it didn't work out nonetheless. And going through, and oh, what's the name? There's that stupid pun about the guy. He's really short, and something something about the tracks being too loose, Toulouse-Lautrec. And suddenly it pops out there, and you've got enough of these inputs coming in there, and this is tip of the tongue wiring. This is how you may not be able to just remember the guy's name. Wait, he was the short guy with the beard who hung out in bars and Parisian bars, and here was that time in 7th grade. And enough of these inputs, and suddenly out pops the information. What this begins to tell you is this is ways of getting similarities. These are ways of getting things that vaguely remind you. This is a world where humans can now do stuff like have a piece of music that reminds them of a certain artist because they both have similar coloration. And that's something that makes sense to us. That's something that can work because what you then begin to see is every one of these neurons, this one for example, impressionist neurons, this one may also be at the intersection of another network that's going this way, a network of French guys from the last century. And it may be part of another network of people whose names are hard to pronounce, so you're anxious about saying them in a lecture. Or the intersection, and each one of these is going to be an intersection of a whole bunch of these. All of these networks, what does that do? That's what you can do that a computer can't. You see similarities, similes, metaphors, and somewhere in there you get something really important, which is the networks that have wider expanses that connect to a broader number of neurons in a very simple, artificial, idiotic way. That's kind of what creativity would have to be. Networks that are spreading far wider than in some other individual. It is literally making connections that neurons in another individual does not. And suddenly you have a world where, everyone knows this one is a face. And it was only a limited number of people who ever decided that this one's a face. And in some level, Picasso had like a different network, a broader one, as to what could constitute a face. A broader network in some way is going to have to be wiring that is more divergent and at the intersection of a bunch of networks that are acting in a convergent way. So what's some of the evidence that it actually does work this way? You go and you stick electrodes into neurons in the cortex, and what you see, if the world was entirely made up of like Hubel and Wiesel, one piece of knowledge only, what you would see is you would find neurons that each one responds to one single thing. All these grandmother neurons. And instead what you see by the time you get to the interesting part of the cortex, past the first 3 layers of the visual cortex and the first 3 layers of the auditory, once you get into the 90%, that's called the associational cortex, and it's called that because nobody really knows what it does, then what you see are neurons that are multimodal in their responses. All sorts of things stimulate them. And here we have a neuron that's being stimulated by a type of painting, by the knowledge of French guys, by something phonetic, by all sorts, and they're multi-responsive. So that's what you wind up seeing. The majority of cortical neurons, when you record from them with an electrode, they're not grandmother neurons, they're at the intersection of a bunch of nets. More evidence for this. This was one of the grand poobahs in neuroscience around the 1940s or so, a guy named Carl Lashley, and obviously a very different time in terms of thinking about specification of brain function. And what he did was a very systematic attempt to be able to show where in the brain individual facts were stored. And the term for it at the time, this jargony term, was engrams. He was searching for the engram for different facts, and what he would show was he would destroy parts of the cortex in an experimental animal, and he couldn't make the information disappear. He would have to destroy broader areas, and some of the knowledge, some of the memory was still in there, and he concluded in this famous paper in the Search for the Engram that according to all the science he knew, there could be no such thing as memory. And the reason why was he was working with a model of being able to, there's a single neuron where if I could ablate it, I should be able to now show in that rat that it's just lost the name of its kindergarten teacher. And instead, you see networks going on. You see the same thing clinically in something like people with Alzheimer's disease. Early on in Alzheimer's, you will lose in these networks, you'll lose a neuron here, you'll lose a neuron there when you're just beginning to lose neurons. And what you see is clinically in people with Alzheimer's early on, it's not that they forget things, it's not that memory is gone, it's just harder to get to. And you do this with all sorts of testing, neuropsychological testing, where you try to give the person cues to pull it out. Example, you're giving somebody, potentially with Alzheimer's, a classic orientation test. You ask them, okay, do you know the name of the president? Okay, they managed to get that. Do you know the name of the last president? No idea. So now you give them a little bit of cueing. Okay, let me help you a little bit. It's a one-syllable word. Still not there, even though you've now activated the one-syllable word network, obviously artificial. Still can say, okay, let's make it a little bit easier. It's things you could find in a park, in a city park. So you're activating that, but no, still not coming out. And then you give even more explicit priming there. You give them a forced choice paradigm is what it's called. So, okay, so is it President Tree or President Shrub or President Bench or President Bush? Bush, the kid with the father also. It's still in there. It was still in there. It just takes more work to pull it out. What you're seeing there is not the death of individual memories. You're seeing a weakening of a network that is now taking stronger priming to pull it out of there. And just to show how subtle network stuff can be, here's something that would work with a lot of individuals with early stage dementias. What you do is another type of priming. So you're eventually going to ask them the name of the previous president, and they first come in and you say, oh, great to see you, come on in, what a beautiful day. I walked here by way of the park. The bushes were so beautiful this morning. In the park there, some of them had flowers, some of them didn't, but bushes are so nice to look at when you're walking through a park because bushes are one of my favorite forms of bonding. And then 5 minutes later, they are more likely to remember the name Bush out of a whole different realm of more subtle networks you're tapping into. So all of this is the beginning of a way of solving the problem we had the other day of not enough neurons for them to be grandmother neurons. More solutions. We then went to our next realm of trouble, which was the problem of there's not enough genes. There's not enough genes in that specific realm of explaining bifurcations, and there can't be a gene that specifies, okay, this is where you bifurcate if you're this particular blood vessel, and a different gene for this particular bronchiole, and a different gene for this branch of a dendrite in a single. It can't work that way. There's not enough genes. What this introduces is the idea of there being fractal genes. Genes whose instructions are ones that are scale-free. What do I mean by this? Okay, here's what a fractal gene might do. So we've got a tube, and remember this is a tube that's going to be part of a blood vessel or a dendrite or a lung or whatever. We've got a tube. And the fractal rule here is grow this tube in distance, grow it until it is 5 times longer than it is wide. The width, the opening, and that's the simple rule. And the rule is when it's grown 5 times longer, bifurcate. So what's going to happen at that point? It's just gone 5 times longer, and it bifurcates at that point. And what you've got is now, because this is split in 2, the cross-section is going to be shorter. But you apply the same rule. Now with the shorter cross-section, you have the same rule, grow 5 times the length of that cross-section until you split. And what you wind up getting is, this is one simple fractal rule that will generate the tree patterns. That the branchings get shorter and shorter, the distances between the branch points get shorter and shorter because the cross sections are getting one simple rule, and you could generate a circulatory system, a pulmonary system, and a dendritic tree by giving a fractal instruction. In this case, one that is scale-free. That is independent of what the unit is here, and this could work within the single neuron or within an entire circulatory system. So all of that's great. That's totally hypothetical. Ooh, fractal genes. We know by now that's got to translate into a protein in some way or other. How might this actually look in a real system? So suppose okay, so a gene coding for a protein. This is one copy of the protein. This is another. This is another. They bind to each other in a way so that they form a tube. and they bind to each other in a way that's just pure mechanical reality of these are not bits of information, these are actual proteins. So it's going up in the tube there, and suppose that the forces are, as the tube goes up, it gets more and more unstable, and when the tube is high enough, it gets unstable enough that these bonds between the proteins begin to weaken, and it begins to split. The splitting there is a function of the length of these. So it's split, and now the next one has half the number of proteins in this one, and thus it's that much weaker. So you only have to go a shorter distance now before it begins to split. This doesn't exist. There's no way it's like this, but what you could begin to see is here's how you could turn a scale-free set of instructions potentially into what it would actually look like with mortar and bricks in terms of proteins, how it might actually work. Now the notion of fractal genetics, of fractal genes and fractal instructions begins to solve another problem. And this is that space problem of how much stuff can you jam into a space. Here's the challenge here in terms of how dense things are. In the body, amazing factoid, there is no cell in your body that is more than 5 cells away from a blood vessel. Okay, you could see why you would want to do that, but that is not an easy thing to pull off. How do you do that with the circulatory system? An amazing other factoid to factor in with that is the circulatory system comprises less than 5% of your body mass. How can this be? You've got this, system that's everywhere, but it's taking up almost no space. It's within 5 cells of every cell out there, yet it's less than 5% of the body. And, okay, forget it, I'm not going to put that up. But what this begins to, okay, you convinced me. So let's do this. So what you begin to do is transition to a world of fractal geometry. You've got all your Euclidean world of nice, smiley, strange things there. You've got this whole world of shapes that are constrained by classic Cartesian geometry and all of that. And what fractal geometry generates are objects that simply could not exist. Here up on top, eventually, you will see the first example of this, and this is out of the Chaos book, and this is this Cantor set. What you do is you start with a line. You start with a line, and you cut out the middle third. Now for those remaining 2 ones, you cut out the middle third. For those remaining 4, you cut out the middle third, and there it is. And you just keep doing this over and over and over again. And what do you do when you take it out to infinity? What have you generated? A set of an infinitely large number of objects, lines, that take up an infinitely small amount of space. It's not possible for that to work, yet as you go more and more that direction, you get this impossible phenomenon of something approaching having an infinite number of places that something appears while taking up almost an infinitely small amount of space. And what this winds up being is, it's not quite a line anymore at the bottom, but it's kind of more than a dot. It's somewhere between 1 and 2 dimensions. It's a fractal. Its dimensional state is somewhere 1 point something or other. It is somewhere between dots and a line, and it does this impossible thing, which is it's everywhere without taking up any space. Or you could then push it to the same thing in the next dimension. And this is this Koch snowflake, and it's the same sort of rule. You start with the triangle there, and the rule is you take the middle third and you put a little triangle out of it. And then you take the middle third of that and put a little triangle out, and the middle third, and you just keep doing it forever and ever and ever, and you wind up with something that is impossible, which is an object that has an infinite amount of perimeter, an infinite amount of surface area within a finite space. That's impossible. But it begins to approach this. And what do you see here? This is a way of just iterating over and over and over to jam a huge amount of surface area into a tiny space. And thus it's somewhere but different, sort of like a line, but it's sort of like a plane by then, and it's got a fractal form somewhere between 2 and 3. It's got a fractal quality of 2 point something or other. It's an impossible object that is solving this problem of being, in another version, having surface area everywhere without taking up any space and being within a finite area. Next, finally, this Menger sponge, which is the same exact concept again. You start with the box up there, the ring, and you take out the middle third of each of those segments, and then you take out the middle third of each of those segments, and if you're doing this with what starts off as a three-dimensional cube, eventually you get something that cannot exist, which is an object that has an infinitely large amount of surface area while having no volume. That's what it produces at the extreme, and we got something here that's somewhere between two different dimensions, a fractal again. And what you see is, this is how the body solves the packing problem, because all you need to do is make the circulatory system, some version of this, some version of splitting the ends of the capillaries over and over and over, or making the lungs with their surface area for exchanging oxygen looking something like this, and this is how you generate a system that is everywhere and taking up virtually no space. Obviously, it's not taken out to infinity, but this is how you can have a circulatory system that's 5 cells away from every cell in the body, yet takes up less than 5% of the body. This is a fractal solution. All you do here to generate these is taking some of these qualities over and over and over and over, and you can begin to produce absolutely bizarre, impossible things in terms of surface area and perimeter and volume and all of that. This is how you can use a fractal system to solve the packing problem. Now, of course, as soon as you're coming up with the notion of something like fractal genes, you, of course, have to consider the possibility of there being fractal mutations. What would a fractal mutation look like? And again, most people, most geneticists and molecular people do not think about this in these terms, but there are people who do, who actually talk about things like fractal gene mutations. What would it look like? Suppose you've got a mutation, and it produces a protein that's slightly different, and as a result, it's got bonds here that are slightly weaker between different proteins. So on a mechanical level, what have we just defined? This is a tube that's going to grow of these proteins where it's a shorter distance before it begins to split, because these bonds between them are not as strong. There's a mutation now where instead of growing 5 times the cross-section, maybe you're growing 4.9 times the cross-section. And thanks to that mutation, the entire branching system is going to be compacted a bit. It's not going to reach the target cells. And these would be catastrophic mutations where the pulmonary system doesn't develop, the circulatory system doesn't develop. And what you would see in those cases is the mutation is something that has consequences that are scale-free. Another hint when you see some fractal gene mutations are a small number of diseases that they're about spatial relationships in the body. For example, there's a disease called Kallmann syndrome where you get stuff that's wrong with midline structures in the body. Something is wrong with the septum between the noses, the nostrils. Something is wrong in the hypothalamus. Something is wrong in the septum of the heart. This is not 3 different mutations. This is some sort of fractal mutation messing up how that embryo did symmetry, how the embryo does midline structures. So you begin to see ways here in which you can solve this, and within biological metaphor where you could begin to get solutions for these problems that are also mutations that can put you up the creek. Okay, so that is another realm for beginning to solve this. Another domain, and here we begin to move into the realm of emergence, emergent complexity, which we will first look at a couple of crude passes at it. First, emergence driven by biophysical properties, and do not freak out if you don't know what I mean, because I have no idea what I mean by that, so I will explain it in a more accessible way. And this was something that was explained endlessly by a guy who used to be in the bio department, a developmental botanist named Paul Green, who died about 10 years ago, way too young from cancer. He was a really good guy. he would give this famous lecture where he would start off and he would describe some sort of disk, and the point is the disk, the material inside was of a softer material than the material on the perimeter, and he'd be putting up math at this point that I didn't understand, but it was sort of a disk like that, and then he would show that what happens if you heat the system. What happens if you put heat on a disk like this? And what he would wind up showing, going through agonizing amounts of math, is that when you heat a system, the only solution for this system that's trying to respond to the heat, but in different ways on the perimeter versus the inside, is to come up with a double saddle shape. And the math proved this, and I had no idea what he was talking about. You come up with a double saddle shape. And then what he says is, so that's how you get a potato chip. You take a slice of potato where there's more resistance on the perimeter and less on the inside, and you heat it, and the only solution to that problem is to come up with a double-saddled potato chip shape. And if you change the outside, the force of it, if you take one of those like Great Organic, give you the runs type potato chips, where it's going to have the skin left on the outside, it's going to be a somewhat different shaped double saddle, because there's only one solution mathematically to that. And then you sit there and you deal with a very simple important fact, which is that slice of potato knows no biophysics. That slice of potato didn't fit. There's no gene that instructs potatoes to respond to heat in this way. This was the inevitable outcome of the biophysical properties of a slice of potato. And what he then shows is in plant systems after plant systems, they develop where two shoots come out this way, and a little higher up, two shoots this way, and two this way, and two this way. They're all double saddles, and this winds up being a mathematical solution to a packing problem there when plants are growing their stems. There's no genes specifying it. You don't need genetic instructions. It is an emergent property of the physical constraints of the system. Another example here that's sort of proto-emergence, somewhat simpler versions, this phenomenon of wisdom of the crowd. And this is one was first identified by Francis Galton, who was some relative of Darwin and started eugenics and was bad news in that regard, but famous statistician. And being an Englishman somewhere in the 19th century, he spent huge amounts of time going to state fairs and county fairs or whatever. And he was at this fair one day where they had some oxen up there, and they were having a contest that if you could guess the exact weight of the oxen, you would get to milk it or something. I don't know what the prize would be. And there were hundreds of farmers around filling out little pieces of paper where they were guessing, and what he discovered at the end was that nobody got the answer right. Good. So the owners of this get off easy without having to give up any of their oxen milk. But he then did something interesting. He collected all the little slips of paper, and he averaged all of them, and it came out to the correct weight within an ounce. In other words, no individual in that group had enough knowledge to be able to truly accurately tell what this thing was, but put them together in a crowd, and out comes the right answer. Another version of this, and this one is deeply important in terms of Western intellectual tradition, back to, is that program Who Wants to Marry a Millionaire, does that still exist? In reruns and, okay, so it was this one, they give you questions, and if you answer them, they give you money, and it's great. And at various points, if you're stumped, you got three things you could do. One is they could eliminate, you got four choices, they can eliminate two of them to make it a little bit easier for you. Another is you have this expert who you can call up. And the third option is to ask the audience which they think is the right answer. And all the audience there has these little buttons, so they could choose A, B, C, or D of the multiple choice there. And what the logic is supposed to be is cut it down to two. Your chances are better if you have to guess. Talk to your wise expert who's sitting by on the phone there, and they're going to be wise and be able to hopefully answer this question, or ask a whole bunch of people. And they would all vote, and any smart contestant would choose whatever the audience chose. Because when the audience was asked, 91% of the time, they got the right answer. They got the majority of people voting for the right answer, and this is more wisdom of the crowd. And this was a much better hit rate than whoever the expert was on the other side of the phone. One person could be extremely expert, but they're not going to be as expert as a whole bunch of somewhat decent experts thrown together. This is the notion behind a field called prediction markets, where what you do is you are trying to predict some event. For example, the Pentagon is very interested in using prediction markets to try to predict where the next terrorist attack might be. And what you do is you get a whole bunch of experts and you ask each of them to think about whatever the parameters are and take a guess as to how long it will be before the next one occurs. And what you do is you average them up and assume there's a wisdom of the crowd thing going on, and that will give you lots of information. Great case of this a few years ago. There was some submarine or something that sunk somewhere out in the Pacific in the ocean, and nobody knew where it was, but they kind of knew where the last sighting, the last recording was from it, whatever. They had a whole bunch of naval experts, and they had all of them sort of bone up on the knowledge of what was the water temperature and wind speeds and where they were in the last sighting and what was on TV that day or whatever. They got all the information, and each one made a guess as to where it would be on the map, and you put them all together, and they had guesses covering hundreds of square miles of ocean floor, and they put it all together, and they came up within 300 yards of the right location. So what we have over and over here is this business of put a lot of somewhat decent experts together on a problem, and they will be more accurate than almost any one single amazing expert at it, under a few conditions. The collection of these partial experts can't be biased, or if they are, they all have to be biased in a random scattering of directions. And they need to really do be somewhat expert. If you get a whole bunch of people off the subway in New York and ask them to guess the weight of the oxen, they are not going to wisdom of the crowd their way into being able to milk the thing afterwards. You got to have people who have some experience with it. And you wind up seeing wisdom of the crowd stuff going on in all sorts of living systems. For example, here is an ant colony, and here's a dead ant, and they're trying to get the dead ant back to the ant colony. And when you look at these things, they know how to get it, or they get some dead beetle or something to eat, and a whole bunch of ants push it over back to their colony. Oh, does each one of them know exactly where they should be pushing? No, what you have instead is each ant has somewhat of the right idea as to where they should be going, and there's more ants that have a reasonably accurate notion, a smaller number that are somewhat off, a really small number that are way out of whack, because in general, ants are kind of experts at finding ant colonies. They're pretty informed, and what you do is you put them all together and you do this vector geometry stuff, and it moves perfectly in that direction, and no single ant knows exactly where the colony is. You've got a wisdom of the crowd thing here going on. Okay, 5-minute break. If you have a chance, could you email me that website so we could post it in the courseworks? It's great. Okay, picking up. So now we are ready to take some of those building blocks, wisdom of the crowd stuff, biophysical potato chips, and begin to see it more formally in this field of emergent complexity. What is that about? What we've already alluded to, it's systems where you have a very small number of rules for how very large numbers of simple participants interact. What's that about? Here's what emergence is about. You take an ant and you put it on a tabletop and you watch what it's doing, and it makes no sense whatsoever. You take 10 ants and do it, and none of them make any sense. You put 100 and they're all scattering around, and somewhere around, I don't know, 1,000 ants or so, they suddenly start making sense. And you put in 10,000 or 100,000 or whatever it is, and suddenly, instead of some little thing wandering around aimlessly, you suddenly have a colony that can grow fungi and regulate the temperature of the colony and all these things, and suddenly out of these ants emerges an incredibly complex adapted system, an adaptive one. And the critical point there is no single ant knows what the temperature should be in the colony, or if this is time to go out foraging in this direction instead of that direction. It all emerges out of the nature of ant interactions. You've got very simple constituent parts, an ant, much like one box that's filled in a cellular automata. You've got very simple rules for how they interact with each other. Ants have, I don't know, maybe 3.5 rules. Don't tell Deborah Gordon in the department, who's an ant obsessive, but that I may be inadvertently dissing the ants, but they have a small number of rules as to how they interact. If you bump into an ant and you do this with the pheromones, you go this way, and if you go that way, and I'm just making it up, they have a small number of rules, and as long as you got a lot of ants doing this, out of this can emerge hugely complex adaptive patterns. And this is what an emergent system is about. Simple players, huge numbers of them, simple nearest neighbor rules, and you throw them all together and out comes patterning, and there is no single ant that knows what the blueprint is, and there's no blueprint. There is no plan anywhere that says what the mature form of the colony should look like. There are no instructions. It is bottom-up organization rather than top-down. And you see all sorts of versions then of emergent complexity built around, again, lots of elements of things with a small number of very simple rules about how neighbors interact with each other. I don't even need that board. Okay. Here we have 2, 4, 6, 8 different cities, or 8 different places where an ant could find good food, or 8 different something or others, 8 different locales. And you were trying to do something efficient, you need to go to each one of them to sell your product or to see if there's good food there or not. You need to go to all 8 of them, and you want to do it as efficiently as possible. You want to find the way to have the shortest possible path to go to all of these places. And this is the classic traveling salesman problem. And nobody at this point can solve it. There's no formal mathematical solution. And by the time you get to like 8 locales, there's like, I don't know, hundreds of billions of different ways you can do it. So you can't come up with the perfect solution, but you could come up with maybe kind of a good, decent one. There's 2 ways you could do it. First is to have an unbelievably good computer that just by sheer force cranks out a bazillion different outcomes, and in each case measures how much you're doing it, and you can get something close to an optimal answer. The other way of doing it is to have yourself some virtual ants in something that is now called swarm intelligence. Here's what you do. You need to have 2 generations of ants. The first generation, you stick them all down, different numbers of them, and they all start off in these different cities, these different locales, and their rule is each one of them goes to another city. Each one of them goes to another destination. But here's the following rule. The ants are leaving a pheromone trail. Pheromone trail, they stick their rear end down, what is it? Head, thorax, abdomen. They stick their abdomen down, and they've got a gland at the bottom there which releases a pheromone and makes a track, a scent track of the pheromone there. And a very simple rule, they have a finite amount of pheromone in there to expend on the entire path they're making. In other words, the shorter the path, the thicker the pheromone trail is going to be. Now what you do is deal with the fact that the pheromones dissipate after a while. They evaporate, and thus the thicker the path, the longer it's going to be there. You now take a second generation of virtual ants and you throw them in there, and what their rule is, they wander around randomly, and anytime they hit a pheromone trail, they join the trail. one way or the other, and they lay down a pheromone trail of their own with their abdomen. They reinforce the markings on this trail. And let 10,000 virtual ants do that for a couple of 100,000 rounds of generations, and they solve the traveling salesman problem for you, because it winds up being the short paths, the more efficient ways of connecting locales will leave larger, thicker trails, which are more likely to last longer, and thus increase the odds that an ant wandering around randomly will bump into it and reinforce it. And what you see is, initially, there will be every possible path, and as you run this over and over, it will begin to fade out, and out will emerge the more efficient ones. You can optimize the outcome doing it this way, just asking virtual ants to do it for you. And this is exactly how ants do it out in the real world. When they're foraging in different places, there is a first wave of them that comes out, and they go to locales leaving scent trails, and then there's the wanderers that come in, and when they hit a trail, they join it. There are now telecommunications companies that use swarm intelligence to figure out what's the shortest length of cable they need to use to connect up, 8 different states worth of telecommunication towers, whatever they're called, and they can sit there and do math till the end of the universe trying to figure out the cheapest way to wire them up, or they can use swarm intelligence. And that's what a lot of them do at this point. It works. What are the features of it? This is not wisdom of the crowd. This is not that every ant knows a solution to the traveling salesman problem, except none of them have the perfect solution, but put them all together and they all get to vote in outcomes. The ant don't know from traveling salesman problems. The ant knows nothing about trying to optimize this. All the ant knows is one of 2 different rules. If I'm walking from one of these to one of these, the longer I walk, the thinner the pheromone trail. Or rule number 2, if I stumble into one of these, I join it and put down my markings there. 2 simple rules, one very simple type of sort of unit of information, and they're an ant, and all you need to do is make sure there's enough of them, and they solve the problem for you. This winds up explaining another thing. How do bees pick a new nesting site? A bee's nest, a hornet's nest, a bee's nest. Every now and then the bees need to leave and pick a new place to live, and how do they figure out the good place? And there's all sorts of criteria of nutrients, and so all sorts of bees go out there, and what they do is they look for food sources. And they look for a place that will have a lot of food. Maybe that's a place to go and move the colony. So we know already the bee will go out and find its food there, its food source, and we'll come back in, and here's the colony cut and cross section. And what you wind up having is this ring of bees. Here's the entry, and you have the bee dancing going on that we've heard about in the middle in the dance floor there. And we've already heard it's this pattern of this figure 8 while shaking the rear end, and we know what the information is, which is the angle tells the direction to go out there. And the extent to which it's wiggling its rear end is how long you're supposed to fly for. But the final variable is the better the resource, the longer you do the dance. So you've got bees coming in from all over the place that have found good resources, that have found so-so ones, all of that. And so there's bees doing all this dancing stuff here of different durations. And the ones who have found the good solution to where do we want to live are dancing longer. The ones who have found the most efficient path are leaving a message longer. So now you bring in your second generation, and the rule is, among bees, if they happen to bump into a bee that's doing a dance, the bee responds and goes where it tells you to go. So a bee may randomly sort of bump into one of these guys, and then off it goes. Actually, I'm sure it's more complicated than this, but it's along the lines of there's now random interactions. If one of the peripheral bees bumps into one of these bees that has information, it joins in that bee's group. And it then goes and finds the food resource and comes back with the information. So thus, by definition, if you have found a great food source, you're going to be dancing longer, which increases the odds of other bees randomly bumping into you. which causes them to go and find the same great food source and come back and dance longer, and the ones with lousy ones are coming in and dancing very briefly, and thus there's hardly any odds of somebody bumping into them. And what you begin to do is you suddenly optimize where the hive is supposed to go. Again, it's not wisdom of the crowd, it is an emergent feature of one generation with information based on some very simple rules, and one information that generates some random element, and out comes an ideal solution. More versions of this. Another domain where some very simple rules, out of it emerges something very complex and adaptive. Okay, so the themes here are 2 generations, the more adaptive the signal, the stronger it is, the longer it lasts, and then a randomization element. Another theme that comes through in a lot of emergence, which is to have your elements in there, your ants, your bees, your traveling salesman, whatever the constituents are, and now what the rules are simple rules of attraction and repulsion. Which is to say, some of the elements are attracted to each other, and some of the elements are repulsed by each other. Some are pulled together, some are pushed apart, like, for example, magnets. Magnets are polarized in the sense that magnets only have 2 ways of interacting with each other, simple nearest neighbor rules. They're either attracting or repelling depending on the orientation. So here's what you do now. You take a system and something very simple. You've got some simulated sim city sort of thing where you're letting out of a system run to design a city. You want to do your urban planning in your city that you're going to construct there. And what you do is you can sit there and you can study millions of laws about zoning and economics and all of that to decide something very simple. Where are you going to put the commercial districts, and where are the residential districts going to be? Or you can have just a small number of simple rules, which is, for example, if a market appears in some place, what it attracts is a Starbucks. And what it also attracts is a clothing store or some such thing. So a bunch of rules, but then you have repulsion rules, which is if you have a Starbucks, it will repulse any other Starbucks. So the nearest other Starbucks can be this far away. If you have a competitor's market, it can't get any closer than this. That sort of thing, these simple attraction repulsion rules. And what you wind up getting when you run these simulations are commercial districts in a city, where you get clusters of commercial sort of places that are balanced by attraction and repulsion, where you have thoroughfares connecting them, and the more elements there are in the 2 neighborhood commercial centers, the bigger the connection is going to be, the bigger the street is, the more lanes, the more powerful the signal coming through there. And you throw it in, and out pops an urban plan that looks exactly like the sort of ones that the best urban planners come up with. And all you need to do instead is run these simulations with some very simple attraction and repulsion rules. So you do that, and it winds up producing stuff that looks like cities. You do that with a bunch of neurons. You take a petri dish and you throw in a whole bunch of individual neurons, and they have very simple rules. They secrete factors which attract some types of neurons, and they secrete factors which repel other types of neurons. And all of them are having some very simple rules. When I encounter this, I grow projections towards where it's coming from. If I encounter that, I grow projections in the opposite direction. Simple attraction and repulsion. And what you do is, at this point, you throw in a whole bunch of neurons, each one, where you throw them into a petri dish, and at the beginning, they're all scattered evenly all over the place, and you come back 2 days later, and it looks just like this. You have clusters of neurons sending projections, and you have all these empty residential areas in between. And if you just mark this in a schematic way, looking from above, you're not going to be able to tell, is this the commercial districts in a big city, or are these neurons growing in a dish? And you get areas of nuclei of cell bodies and areas of projections, and it winds up looking exactly like that. And amazingly, there was a paper in Science earlier this year, and it was looking at one of these versions again, in this case attraction and repulsion rules with ants colonies setting up foraging paths, and they explicitly compared one colony to the efficiency of the distribution of the train stations in the Tokyo subway system. And what they showed was very similar solutions, but the ants had gotten a more optimal one. And the subway system had people sitting there salaried to figure out the best way to do it. All the ants had were very simple rules of, if it's someone from the other colony, I stay this way. If it's someone from mine, simple attraction and repulsion, and out comes something that looks like this as well. So here you see that happening with a remarkably small number of rules. Now you put it into a really interesting context, which is something we bumped into back when first introducing proteins and DNA sequence equals shape equals function, all of that. Molecules have charges on them. Some of them were positively charged, some of them are negatively. Whoa, attraction and repulsion. Positively charged molecules are attracted to negatively charged ones, same charged ones repulsed. Here we have a system with very simple attraction and repulsion rules. And that's the logic behind, when one thinks about it, one of the all-time important experiments, something that was done in the 1950s by a pair of scientists, University of Chicago, Urey and Miller. Here's what they did. They took like big vats of organic soup stuff that just had all sorts of simple molecules in there. Little fragments of carbon to carbon, or little fragments of all sorts of inorganic molecules, and there are little ones in there floating around in this organic soup. And what they did was they would pass electricity through it. And they did this vast numbers of times. And eventually, what they saw was they would come back and check, and these random distribution of these things, of these little fragments, had begun to form amino acids. Whoa! Metaphor. The organic soup, just the evenly distributed sort of world of potentially organic molecules in a world in which electricity passes through. Lightning, what had these guys just come up with some in your like kitchen sink experiment of the origins of life? And what people have done subsequently is show you don't need the catalyst. There's a whole world of researchers who study origin of life, and the basic notion is you put in enough simple molecules in there that have attraction and repulsion rules, and you get perturbations and spatial distributions of certain ways, and they will begin to form rational structures after a while. Here's another version of this, and I used to do this in class, except I can never pull this one off, and it just became chaotic. It's a kid's toy. You got these magnets. You either have magnets like that, and then you have little metal balls that can go onto the magnet here, and you've got vast numbers of them, and you can piece them together. Whoa, this is starting to look kind of familiar here. So we have these constituents with very simple rules, which is the magnets repel each other. They bind these things, and here's what you would do. Here's what I would attempt to do. First off, I would get somebody to show me how to get the video thing on here to project it, but you would put up a whole bunch of these magnets in rows, not too close to each other, nice and symmetrical, and what you do then is you take a handful of the metal balls and fling them in there. And if you do that 400 or 500 times, eventually they will bounce around, and amid all the pieces flying, you are going to get a pyramidal structure like this. One of those, just like that, it's three-dimensional, you know, that you're going to get one of those that will simply pop out of this, because that's the nature of potato chips solving their math problem with double saddles. That's the nature of throwing a whole bunch of elements with simple attraction and repulsion rules, and give them enough chances, throw in enough perturbations there, and structures will begin to emerge. And it's the same exact principle there, these same ones over and over. So we've got some very simple versions where you get emergent complexity. One is this version of a first generation has directed searches, and the intensity of the signal that it leaves afterward is a function of how good of a search they've done, random wanderers. Then you have the attraction-repulsion world of putting these together, lots of elements, and you begin to get structures out of it. Next version of this, or next domain of where you begin to see the fact that these rules are underlying an awful lot of things. Suppose here you are studying earthquakes. And apparently there's just like little earthquakes going on, 20 times an hour or so, all down in the Richter scale of, 1/4, or who knows what. But you get enough of these, you get a huge database, and you can begin to graph the frequency of Richter 1 earthquakes, and how often do you get the Richter 2 and Richter 3 and all of that, and you graph it, and it's going to look something like this, a distribution like that, which is obviously there's a huge number of number 1 categories, and it drops off until the extremely rare at this end. There's a distribution which is mathematically can be described something called a power law distribution with a certain angle to it. And okay, so here's the relationship between how often you get little teensy earthquakes and the big ones. Now instead, what you do is something much more different from that, which is you look at 50,000 people and you look at their phone calls over the course of a year, and you keep track of how far the phone call was, how distant the person is that they called. And now you map the distance, the very shortest calls, the very longest, and the frequency, and it's the exact same curve. It's the same power law distribution. Next version of it, this was a study that was done, which was, I don't quite know how these guys did it. I always get lost in the math on these, but in this one, what they did was they took a whole bunch of marked dollar bills, and they started it in the middle of, I don't know where, I think it was Columbia, something, and they were somehow able to keep track of how far the bills had traveled a week later, and asking, okay, how many of the bills had traveled no more than a mile, how many 5 miles, how many, and it was the exact same curve. And people now have been showing this same power law distribution. Here's some of the things that have been shown. The number of links that websites have to other websites. The number that have only one link, the power law distribution. Proteins. The number of proteins showing certain degrees of complexity, and the numbers dropping off with the same power law. Here's one, which is the number of emails somebody sends over the course of the year. This is the one that was done at Columbia. They got access to everybody's email records. I don't understand how they could have done this, but it was a couple of million over the course of the year. And what they showed was the frequency, how many people were making this small the number of emails over there, and the same power law. Then there's this totally crazy one, which is, okay, do you guys know the Kevin Bacon 6 degrees of separation thing there? Okay, someone went and did a study about this, that they got like every actor that they could find who was in a film in the last 2 years, and they got all of their filmographies, and they generated their Kevin Bacon degrees of freedom, degrees of sing it out. Okay, and they figured it out, the number for each individual, and then they graphed it. How many people were 6 degrees of separation away, how many were 5, so on, and it's the same pattern. And this one keeps popping up, this power law business, and what you see intrinsic in that is it's a fractal. Because some of the time you're talking about what's happening with the tectonic plates on Earth, and some of the time you're talking about phone calls, and some of the time you're talking about how molecules interact with each other. There's something emergent that goes on there, which is an outcome of some of these simple attraction-repulsion rules, an outcome of simple pioneer generation and then random movement ones, and outcome structures like these. This winds up being applicable in a very interesting domain biologically. Okay, so now we go back to the traveling salesman problem, and we're having now a cellular version of it in terms of networks. You've got a whole bunch of nodes here, and the choice that each node has to make, in effect, is how many connections it will make in the network to other nodes, and how far should those connections be. Should it only connect with one's way out there? What does it want to do? That's nonsense in terms of optimizing a system. What do you want your distribution of connections of nodes in a network to be? What is it you want to optimize? You want to get a system that has very stable, solid interactions amongst clusters of nodes, but nonetheless occasionally has the capacity to make long distance connections there. And what you wind up seeing is if you generate a power law distribution in terms of, okay, all of my projections are going to be within this distance, and this then the same power law distribution, so that the vast majority of the nodes in the network are having very local connections, but still there's a possibility now and then of very long ones. You get a system that is the most optimal for solving problems, most cheaply in whatever the term is there, and this solves it for you. And then you look at brain development. So you've got neurons forming in the cortex, in the fetal cortex, and you've got neurons. You've got all these nodes, and they have to figure out how to wire up with each other and how to wire up in a way that is most efficient. What's most efficient in order to be able to do the sorts of things the cortex specializes in. And you now begin to look at the distribution of projections, and it's a power law relationship. Most neurons in the cortex are having the vast majority of their projections very local, but then you have ones now and then that have moderate ones, even rarer ones that have extremely long ones, and you look, and this is how the cortex is wired up. It follows a power law distribution, and what this allows you to do is have clusters of stable functional interactions, but every now and then you can talk to somebody way over at the other end of the cortex to see what's happening. Interesting finding, autism. Autism, people have been looking for what's up biologically, and the initial assumptions would be there's not going to be enough neurons in some part of the brain, or maybe too many in another. What appears to be the case so far is there's a relatively normal number of neurons in the cortex. But then some people started studying the projection profiles of neurons in the cortex of individuals with autism post-mortem, very rare to get these, and you see a power law distribution, but it's a different one. It's a steeper one. What does that mean? In the cortex of autistic individuals, way more of the connections are little local ones. There's far fewer of the long distance ones. There are way more local ones. What does that produce? Little pockets, little modules of function that are isolated from other ones. And that, in some ways, is what's going on functionally in someone with autism. There's a lack of integration of a whole bunch of these different functions there, and that's what happens when you have maybe a mutation or maybe some epigenetic something or other prenatally that changes the shape of the power law distribution. Interesting. There's a gender difference in the power law distribution of wiring in the cortex, which is, in the typical female brain, if this is the power law distribution, and in a male brain, it's a little steeper. Male brains are more modular in their wiring. What's the biggest part of the brain? Okay, we're running out of space here. There it is. There's the brain in cross-section, and you've got cortex here and cortex there, and famously, here's all the cell bodies, and when projections are going from one hemisphere to the other, it goes across this huge bundle of axons called the corpus callosum. The corpus callosum is thicker in women than in men on the average. It is thicker in females than in males because the power law pattern is such that there are more long-distance connections in female networks, and thus it's a thicker corpus callosum. The same thing is playing out with connections like this and connections, but this is the big honker one. You get a thinner corpus callosum in men. You get an even thinner corpus callosum in people with autism. Again, that hypermale notion there of Baron-Cohen's. What you have here is perfectly normal number of neurons, probably even perfectly normal number of connections between the neurons, but they're more local, they're more isolated in the autistic cortex. There's less integration of function. It's more isolated islands of function there. Okay, more examples of where you can get sort of patterns coming out. Another version of it, which is bottom-up quality control. You start a website, you are selling some product, you are selling books or whatever, and you're asking people to rate the books, and you have a board of experts that read all your books, and there are editors, and they're wise, and they're learned, and they write your book reviews and recommend which one should be bought and which one's not, and you get this very successful business going so that you're selling more and more different kinds of books, and as a result, you need to hire more and more of these experts to read the books and produce their ratings, and evalually that just becomes too top-heavy. And what do? The whole world that we completely take for granted now, you have bottom-up evaluations. Everybody rates things, and that's the world where you punch in a book into Amazon, or you look at something in Netflix, and when you return it, it will give you people who liked this movie tend to like these things as well. There are no critics, professional critics, sitting there doing top-down evaluations. This is another realm of expressing attraction and repulsion rules. I liked this, I didn't like this, and all you need to do then is throw in elements of randomization, and you've got bottom-up quality control. And that's a completely different way of doing these things. What's the greatest example out there of bottom-up systems with quality control? Wikipedia. Wikipedia does not have gray-bearded silverback elders there writing up the Wikipedia knowledge and sending it on down to everyone else. It is a bottom-up self-correcting system. It is very easy to make fun of some of the stuff that winds up in Wikipedia, which is like wildly, insanely wrong. But when you get into areas that are fairly hard-nosed, very interesting study about 5 years ago that Nature commissioned, which was getting a bunch of experts to look at Wikipedia and to look at the Encyclopedia Britannica and look at the hard-nosed facts in there about the physical sciences, the life sciences. And what you got was Wikipedia was in hailing distance of the Encyclopedia Britannica's level of accuracy. And that was 5 years ago, and it has 5 years of self-organized correction since then. This is amazing. The Encyclopedia Britannica is like written, there's like 30 like elderly stuffed British scholars that they like have locked in a room for years who produce the encyclopedia, and these are the lawgivers and the knowledge, and you just let a whole bunch of people loose with somewhat differing opinions about whether Madonna was born in 1994 or 1987 or whatever it is, and you throw them all together and you do wisdom of the crowd stuff, and out comes a self-correcting, accurate, adaptive system with no blueprint, just with some very simple local rules. Very simple ones, which is looking for similar patterns shared between different individuals and self-correcting. Where you get even more efficient versions of that is with a lot of websites where not only does everybody get to put in their opinion, but people whose opinions are better rated have more of a voice in evaluating somebody else. You're putting in weighted wisdom of the crowd type functions in there, and out comes incredible accuracy. These are great. There's one drawback with those systems though, which is with ones like Netflix, where it tells you you're going to like this if you like this, that sort of thing. It's a system that's very biased towards conformity. It's not good at spotting outliers of sort of taste and such. What you really want to do in those systems is, here are the movies that are out right now. Here are the ones that have 10% of the people think it's the greatest movie they've ever seen, and 10% think it's the worst movie. That's an interesting movie to see. That's when you want to be able to get away of bottom-up information about the extremes, movies that generate controversial. Everybody's going to love whatever it is, and that doesn't take a whole lot. This is a way to break the potential for conformity in these bottom-up systems. Nonetheless, overall, it winds up solving a problem without professional critics, without a blueprint, without top-down control. So how do you wire some of these up? Back to the cortex and the adult cortex, and has these power law distributions, and they're great because they optimize. They've got lots of stable local communication, but there's still the ability to do creative long distance connections. So that's great. But how do you get that? How does the nervous system wire up this way? And it does swarm intelligence. The developing cortex does a swarm intelligence solution. When the cortex is first developing, what you will have is a first generation, a pioneer generation. A pioneer generation of cells, the cortex surface, all of that there's a pioneer generation of cells that basically grow processes up like these. And these are called radial glial cells. What they are, they're the ants with the first generation of setting down the trail here. They're the first bees coming in, and what you then have, the neurons, are the second generation random wanderers, and what they do is they come in, and as they begin to develop, they have rules that when they hit a radial glia, they grow up along it. They migrate along it, they throw up connections, and you do that with enough of the cortex, which is hundreds of millions or billions of neurons in there, and you get optimal power law distributions. All you need are some very simple local rules, and out of that emerges an optimally wired cortex. And it's the same simple emergent stuff going on. Okay. So how do we begin to really apply this stuff to humans? Because it winds up being very pertinent in making sense of some of the most interesting, complex things about us. So, what's the difference between humans and every other species? Nothing all that exciting. From a neurobiological standpoint, you've got this real challenge, which is you look at a neuron from a fruit fly under a microscope, and you look at one from us, and it's going to look kind of the same. Looking at a single neuron, you can't tell which species it came from. We have the same kind of neurotransmitters that a worm uses in its nervous system. We've got the same kind of ion channels, the same sort of excitability, the same action potentials, minor details are different. We have not become humans by inventing new types of brain cells and new types of chemical messengers. We have the same basic off-the-rack neuron that a fly does. Oh, we have very similar basic building blocks. What's the difference, of course, is we've got 100 million of them for every neuron that you find in a fly brain. And out of that comes emergent properties. Great story. Garry Kasparov, I never remember which syllable to emphasize, grandmaster, Russian chess grandmaster in the 90s, and apparently he's rated as one of the strongest of all times, and he was the person who wound up participating in this really major event, which was this tournament with this chess playing computer that IBM had built called Deep Blue or Big Blue or Old Yeller, or what was it called? Deep Blue. Deep Blue. Deep Blue, and they played against each other, and apparently what happened was in the first game, Kasparov won perhaps, and the computer was able to modify its strategy and then proceeded to mop the floor with him. And this was a landmark event in computer science. This was the first time that a computer had beaten a chess grandmaster. Amazing event. Not surprisingly, afterward, Kasparov is all bummed out and depressed, and his friends were trying to make him feel better. And they go to him and they say, look, all you got done in by is quantity. All you got done in by is the fact that that computer could do a whole lot more computations than you could in a set amount of time. I'm told apparently chess grandmaster types can see 5-6 moves ahead, and they can intuit where the interesting ones was, and Deep Blue could calculate every single possible outcome like 7-8 moves in advance, and every time it would simply pick the one that was the best outcome. It was like generating solutions to the traveling salesman problem. Kasparov didn't have a chance because the computer could simply generate enough solutions to pick the right one. So all of them are saying to him, you should not be depressed because all that computer had going for it was quantity. And what he said in response was, yeah, but with enough quantity, you invent quality. And that's the exact equivalent of one ant makes no sense and 10,000 do. That's the exact equivalent with enough of these elements here you optimize. We do not have fancy neurons that are different than in any other species. We've just got more of them. and simple nearest neighbor rules, and you throw, you know, 1 million of them together and you get a fruit fly, and you throw 100 billion of them together and you get poetry, and you get symphonies, and you get theology, and you get all of that, and it's the same building blocks. With enough quantity, you invent quality. And this is the punchline that came out of really important work a few years ago. Okay, we're now, what, 10 years, I think, into having the human genome sequenced. And about 5 years ago, they sequenced the chimp genome. Soundbite everybody learned from whenever back when is that the human and chimp shares 98% of its DNA. So finally, you had these two gigantic rolls of printout, and here's the entire human genome, and here's the entire chimp one, and somebody could finally sit there and compare them and see, indeed, is it 98% shared? And that winds up being the answer, even though what that number actually means is debatable. But that brings up the question, of course, what's the 2%? What's the 2% that differ? And what has come out of that have been some very interesting findings, some that were mentioned earlier on, which is they are disproportionately coding for transcription factors and splicing enzymes, and okay, that amplification of network stuff, it is preferentially coding for non-coding regions, differing but non-coding, all the stuff from back that lecture for getting macroevolutionary changes, that's how you get a different species coming out. But how about other types of genes? What were some of the key differences? Here was one big difference. We have about 1,000 fewer genes for olfactory receptors